all these stuff that we know about your proteins, we came to know them because of protein purification. So we won't be able to determine the amino acid sequence of a protein. We won't be able to elucidate a protein's structure if we don't have a pure sample of the protein, right? So it is important that you know something about protein purification in case you are going to do research that involves such in the future. So protein purification, from the name itself, your aim is to get a pure sample of one protein. So you isolate this single protein from a complex matrix, which is your cell. So after you have obtained a pure sample of your protein, then that's the time that you can study it. You can study its functions, and you can also study its structure. Way back when people were able to develop methods to purify proteins, it was actually quite easy. That's because they were purifying or isolating relatively stable, small proteins. But in these times, the proteins that are not yet studied, most of them are membrane proteins, which is actually difficult or challenging to isolate because of their natural environment. They are embedded in the cell membrane. And other proteins that are relatively unstable, even inside the cell. So these are the challenges faced by um, protein purification experts. Now, protein purification involves several major steps. So, the first step is you select your source. Will you obtain it from a natural source, such as your chicken liver? Next, you will homogenize your source. So, this homogenization step allows you to break the cell membrane or the cell wall if your source is plant. This will release the contents of the cell, thereby bringing you closer to your target protein. After homogenizing, then you can isolate your target protein. You can quantify, so how much of the pure sample you have, and you can now perform your structural or functional studies. So, in this discussion, we will be focusing on the first three steps. So in selecting a source, you can go natural or synthetic. In most of protein purification protocols, okay, natural sources would include livers, heart, or spleen. So you get your proteins from the organs of your living organisms. You can actually also go synthetic. So this technique is what we call recombinant protein production or protein expression. So we use several vehicles to produce our protein. We can use bacteria such as E. coli, yeast, or mammalian cells. So this is a method wherein you have what you call a vector. So the vector is a circular DNA. So you insert the DNA sequence for the protein that you want to be produced inside your cell, or what we call expression system. So you have the vector, you insert the sequence of your protein. So after you inserted this DNA sequence, you place this vector inside your expression system. So you place it inside your E. coli. If you choose to use E. coli, you place this vector inside your yeast cells or your mammalian cells. And then you induce your expression system to produce the protein using this vector, because this vector contains the DNA sequence of your protein, right? So you induce your expression system to produce the protein, and then you proceed to getting your protein from this expression system. So when you select a source, you can go natural or synthetic. Now, homogenization, again, this is a process wherein if you are using tissue samples, or so you have natural source of your protein, you release the cells from the integrity of the tissue. Okay, so it's breaking the tissues apart, so you are down at the cell level. Now, if you go synthetic, homogenization is also the step that you do to destroy the cell membrane of your expression system. So, your E. coli cell, yeast cell, or mammalian cell. So, the end product of this step is called the homogenate. So, you have the tissue homogenate. However, if we go to synthetic, if you choose to go the synthetic path, this step is not called homogenization. We call it lysis. So it's because your source or your expression system is composed simply of cells. You don't have tissues there. So you just have cells. So you don't need to homogenize the system. You have cells. So what you just do is lyse the cells. So lysis of the cells will release your cytosol plus all the other contents. So your expressed protein is found in the cytosol. So breaking the cell membrane will release your cytosol that contains your produced protein. Now, lysis, there are many methods to perform this, and it depends on your expression system. You have lysis methods for E. coli, for yeast, and for mammalian cells. So it's because these cells differ in the integrity of their cell membranes. So for E. coli, one of the common methods to lyse them is sonication. So this is called a sonicator, and you have here the interface where you input the parameters. You have your container with the E. coli plus the culture, of course. So what you do is you place this inside this box, and you have a probe there, right? So you place the probe inside the container containing your solution of E. coli cells. And then when you press on start, the end of this probe will create pulses. Okay, so these pulses, it creates vibrations through the solution containing your E. coli cells. And these vibrations cause the cell wall and the cell membrane of your E. coli to break. Now, if you have a lot of E. coli cells in your system, in your sample, then you can go for French press. So the French press also uses pressure to break the cell wall and the cell membrane of your E. coli. Another way to lyse your cells is through enzymes. So some of the enzymes used in enzymatic lysis are your lysozyme, zymolase, and lyticase. Lysozyme is often used together with sonication for E. coli. You can also combine these methods with freezing and grinding. So what you do is you freeze your E. coli cells first, and then right after the freezing, you bring them out. So in their frozen state, you bring them out of your freezer and then you grind them. So this mechanical action also destroys the cell wall and the membrane of your E. coli cells. Another method is glass bead vortexing. So this is particularly used if your expression system is yeast cells. So you have your yeast cells and you place your glass beads together with your yeast cells and then vortex this sample. So the glass beads also produce physical, mechanical action as you vortex. The glass beads are the ones destroying the cell wall and the cell membrane of your yeast cells. Next, we have what we call the Downs homogenizer. So this one is used for mammalian cells. So mammalian cells are only composed of cell membrane. So this Downs homogenizer introduces very gentle pressure. In fact, it is shearing. It causes shearing on your cell membrane of mammalian cells. So you have this container. This is where you put your solution of mammalian cells, and you have the plunger. So this is made of glass. So you just plunge up and down your Downs homogenizer to destroy the cell membrane of your mammalian cells. And of course, you can use detergents. This one is mostly used for mammalian cells. So you have the RIPA lysis buffer. So detergents are molecules that can insert in the cell membrane. So they destroy the piece of your phospholipids in the cell membrane, causing the cell membrane to just open so that the cytosol can go out of the cell. So all of these lysis methods can be classified whether they are mechanical, ultrasonic, you lyse by pressure, you lyse by temperature, you remember freezing and grinding, or you use chemicals such as your enzymes. Now, in some protocols for protein purification, they combine more than one of these lysis methods. So this is to ensure that the membranes, cell membrane of all cells in your sample are destroyed so that you get the maximum yield of protein. Now, after you homogenize or lyse your cells, of course, we need to remove unwanted debris, such as your cell wall or cell membrane, from your homogenate or lysate. So we do this through centrifugation. So in the lab, you will find your tabletop centrifuges, which are very small, and then you have the medium-sized centrifuge machines, and you also have large centrifuges. This one even has a vacuum, so it creates a vacuum inside the compartment where you place your samples to be centrifuged. So the vacuum is created so that you can spin your samples at a greater RPM. So sometimes these big centrifuge machines can go up to 40,000 RPM or revolutions per minute. With centrifugation, you can also perform what we call fractionation. So in fractionation, you subject your sample into different RPMs. And for each step, you are separating some parts of your homogenate or lysate. So after each step, you get one fraction. So you centrifuge at a specific RPM, get the fraction. For the remaining part of your sample, you centrifuge again at a higher RPM, get the fraction. Centrifuge again at a higher RPM, get another fraction. In each of these fractions, you will have different um, compositions. So an example is what we call differential centrifugation. So after you have your homogenate, so you can filter it so that you can remove unbroken cells, connective tissues, etc. And you have your homogenate without the debris. So if We centrifuge them at 600 gravitational force for 10 minutes. You will see your nuclei being deposited. So that's if you want to isolate something from your nucleus, then you need to perform this step. Now, if you get this fraction and then centrifuge it again at a higher force, so 15,000 G for 5 minutes, you will see your organelles, mitochondria, chloroplasts, lysosomes, etc., being separated. Again, you get this fraction and centrifuge at even greater force. What you will get as precipitate at the bottom are your plasma membrane, microsomal fractions, like fragments of your ER and polyribosomes. Again, get the fraction, centrifuge at even higher force, you will get your ribosomes. And finally, you get your pure cytosol. So this is performed only if you want to isolate some protein or other biomolecule in a specific organelle. For example, you want to um, purify your polymerase, which is found in your nucleus. So you need to isolate your nuclei. So once you have your cytosol, so you lysed your cells, you have your cytosol. So in the solution containing your cytosol, you have your target protein floating there. How can you catch your target protein? So there are several methods to isolate your target protein. The oldest method is the salting out method. So this is based on the solubility of your proteins. So we use salts, but not all salts can be used. Okay, so there are certain characteristics of your salts that allow them to be used in the salting out method. So the principle here is that at low concentrations of your salt in solution, they actually promote solubility of your proteins in solution. So it's because for the specific concentration of salts, they form electrostatic interactions with the charged residues of your protein. And these salts or ions actually promote repulsion of your proteins. So they repel each other. However, if you try to increase the concentration of your salt, this is where you will observe the salting out. So what happens is that if you have the solution with just protein dissolved in it, your water molecules will hydrate your protein molecules. So you have your protein molecules surrounded by water molecules. But if you introduce a lot of your salt, so represented by the red rectangles, these ions compete for the water molecules. So if you have a lot of salt or ions in your solution, a lot of your water molecules will tend to hydrate these ions. So they will leave the protein molecules and go to your ions. So what happens is that you have the proteins by themselves without the water molecules hydrating them. And the proteins will now clump together, they will aggregate, hence they will precipitate or what we call salt out of the solution. So the consequence if you add a lot of salt in your protein solution is that all of the water molecules will go to the ions from the salt and then the proteins will aggregate. Okay, so you will observe a precipitate in the solution. That precipitate is your protein. Now, in our activity in the demo video, the salt used is ammonium sulfate. So again, we don't use any random salt. So why ammonium sulfate? It's because it has high solubility in water, it has minimal temperature effects, and it doesn't react with your proteins. So it doesn't destroy the structure of the protein, it doesn't destroy the peptide bonds. In fact, in what we call the Hofmeister series, uh, ammonium sulfate is one of the top salts that can be used to induce salting out. So this Hofmeister series gives us the relative abilities of cations and anions to induce salting out. So the cations in these series include ammonium, potassium, lithium, magnesium, and calcium. And your anions, you have fluoride as the top, but fluoride can actually react with some of your proteins. So we choose the next one in the list, sulfate. That is why we have ammonium sulfate. The rest, we have dihydrogen phosphate, acetic acid, chloride, nitrate, bromide, and so on. Now, this salting out method can be combined with what we call fractionation. So, fractionation by salting out. So, what is done here is you slowly increase the concentration of your salt. In the process, what you do is you isolate some proteins stepwise. So let's say at 46% ammonium sulfate, this specific protein will salt out. But at a higher percentage, let's say 64% ammonium sulfate, another protein will salt out. So that is fractionation by salting out. Other methods which are not covered in the demo video and I would like to mention to you, we have dialysis. So in dialysis, you have a membrane. It looks like a plastic, but we call it a membrane or dialysis bag. You place the solution of protein inside and then you place this in a solution of buffer. So the outer container is simply buffer. Your solution of protein is inside the dialysis bag. Now, your dialysis membrane has pores, and these pores have sizes. So we call it the molecular weight cutoff. So, for example, if your dialysis membrane has a cutoff of 10 kilodaltons, it means that any protein with a molecular weight greater than 10 kilodaltons stays inside the membrane. The rest of the proteins that have molecular weights less than 10 kilodaltons, they can go out of the membrane, they can go out of the dialysis bag. Okay? Hence, you separate these small proteins from other proteins. Okay? If your if your target protein has a molecular weight of, let's say, 35 kilodaltons, then it's safe to use 30 kilodalton dialysis membrane. So what happens is that other proteins smaller than your target protein go out of the dialysis bag. So your target protein stays inside. Of course, there are other proteins bigger than your target protein. So this dialysis can be combined with other isolation methods, such as gel filtration chromatography. So in gel filtration chromatography, you separate your proteins according to their size. So large molecules or large proteins are the first ones to go out, followed by medium-sized proteins, and the last ones to go out are your small proteins. So it's because the small molecules spend some time inside your um, gel filtration beads. These are literally beads. So it's because they are small, so they can pass through the small spaces inside these beads, while the large molecules, since they cannot pass through the small spaces, they just go straight out of your column. You can also perform ion exchange chromatography if you know that your protein molecule is charged. So these in this chromatography technique, you have beads whose surface are charged. So the beads can have negative charges or positive charges. So if the beads are negatively charged, then it means your protein should be positively charged so that they can interact. Okay? If your beads are positively charged, then your target protein should be negatively charged so that they can interact. We also have what we call affinity chromatography. So this is usually combined with a method. So what you do is you have your target protein, right? You express this in your expression system. So we are talking about the synthetic approach. You express this in your synthetic system. Now, since it's synthetic, it's possible to introduce another protein attached to this protein, right? So you can introduce this protein, we call it a tag. The tag is not limited to a protein. It can be simply a sequence of amino acids, for example. Now, this tag is combined with affinity chromatography. Okay? The tag has affinity with specific beads. Okay? So you buy a specific beads for this specific tag. Okay? Then, if you run your solution of protein into this column, your tag will now bind to the beads because it has affinity. Now, since only your target protein has the tag, other proteins just flow out of the column because they don't have affinity with your beads. So, this is your affinity chromatography. It is actually the most often chromatography technique used in recombinant protein purification. Now, these steps from the expression of your protein, if you go the synthetic path, okay, down to purification, we must be aware of the conditions. We must control pH, temperature, because these two factors affect the structure of your proteins. We must prevent the presence of enzymes that can attack your target protein. We must also prevent the protein to adsorb to surfaces. So very long time inside plastic containers, for example, can lead to loss of your protein sample. And of course, you must consider whether you want to store your protein sample for a long period of time or if you can directly proceed to your structural or functional studies. And while we are going through each step, it is also helpful if you monitor the amount of protein that you are obtaining. So, from the lysis step, so you have the lysate, right? You get a certain sample, a small volume of your lysate, and you check the absorbance. We know that your proteins absorb at 280 nanometers due to your tryptophan, phenylalanine, and tyrosine residues. So you can monitor the amount of protein that you are obtaining each step along the way. You can also check the purity of your protein via the 260 to 80 ratio. So 280 nanometers, this is where we monitor our proteins. 260 nanometers, this is where we monitor your DNA. So sometimes when we try to purify proteins, some DNA can contaminate our sample. So it helps if you monitor the 260 to 80 ratio. So if this is low, so if this ratio is low, it means your proteins are safe. You can be confident that you don't have DNA in your protein sample.